Welcome back everyone to Exploring the Coast with Hannah. On this week's episode, we're going to be looking at the different methods and protocols scientists use to collect data in the field. Let's go. What is a transect? A transect is a line of study with a defined length. Scientists will count species along this line at given intervals. Transects are commonly used to measure biodiversity, distribution, and population abundances of plant and animal communities. The Limpets program at the PG Museum uses transect sampling data collection in both our Rocky Intertidal and Sandy Beach programs. There are three common ways scientists use transects to gather Rocky Intertidal data. Those are vertical transects, permanent transects using random samplings, and permanent transects for total count data. A vertical transect is a line along which data is collected that stretches from the high intertidal zone to the low intertidal zone. Scientists often use bolts to ensure that they revisit the same spot each time they monitor. Monitoring takes place using quadrats at regular intervals along the transect. Vertical transects are used to reveal zonation and changes in species distribution and abundance. Permanent areas using random samplings help scientists track changes in abundances over time. Scientists will treat a large flat area of the Rocky Intertidal like a coordinate plane, with an x and y axis, and put the quadrats down at random coordinates to estimate abundance and biodiversity for the whole area. Permanent areas for collecting total count species data help scientists track key species over time. Species like anemones, owl limpets, abalone, and ochre sea stars all play vital roles in the intertidal ecosystem and can help detect changes occurring within them. For sandy beach monitoring, scientists can use multiple transect lines to gather species data, like the Limpets program does in gathering data on sand crabs. The long pink transect line parallel to shore defines a specific area of the beach where the sampling occurs, and the numbers represent the meter mark on the transect line. The other 5 yellow transects perpendicular to the shore at the designated meter mark are the lines where the samples will be taken at designated intervals. What is a core? A core sample is usually a cylindrical piece of material that is used to take sediment samples. The sizes of the cores can vary in diameter and length depending on the type and the amount of samples scientists need. The Limpets program at the PG Museum uses clam digging cores made of PVC pipe to take sand samples to find, count, and measure sand crabs for our Sandy Beach program. What is a quadrat? A quadrat is a standard unit of area that is used to represent a subsample of the distribution of species over a large area. Since it would be very hard for scientists to count every single plant or animal over a large area, quadrats are an easier method and more accurate way to collect data. Quadrats are usually square in shape, but also can be rectangular, circular, or irregular depending on what kind of data scientists need to collect. For the Limpets Rocky Intertidal program, we place quadrats over a transect line at different intervals depending on the length of the transect. These transects extend from the splash zone to the low zone area of the intertidal. Now I'm going to teach you how you can count species using a quadrat. For the Limpets program at the PG Museum, we have 2 ways that we count species within a quadrat. The first one is counting all individual species within a quadrat. So that's to say, where if we found chitons, we would have to count every single chiton that we found within our quadrat. The second way we count species within a quadrat is by counting the number of squares where the organisms are found or attached. Today we're going to be counting how many squares that we find sea mussels in. Sea mussels are found in the mid-zone area of the Rocky Intertidal and can be found in very dense beds. So this species makes it hard to count all the individuals of, since there can be so many just within a quadrat. Here we have a quadrat with 25 squares that's laid over a mussel bed. What I want you to do is take a few moments and count how many squares have mussels in them. And remember, you don't count anything outside of the quadrat, only what's within the quadrat. The same for the individual squares. Only count squares that have mussels attached within them. So how many squares did you find where mussels are attached? Let's take a look. So as you can see here, there are 23 squares where there are definitely mussels attached. Now let's take a closer look at those two open boxes. Sometimes this gets a little tricky for students or anybody who is collecting data. Remember I talked to you before about we only count squares where something is attached, whether that be the animal or a plant. And as you can see on those two squares, there aren't actually any mussels attached. While there are some mussels, the tops of the mussels to be exact, that are laying over into the square, they're not actually attached in those. So we only have 23 squares where mussels are present. If you counted the correct number of squares where mussels are attached, give yourself a pat on the back. You just collected data the same way a scientist does. Thank you so much for joining me today. Don't forget, a new video of Exploring the Coast with Hannah drops every Tuesday at 11 a.m. on the Pacific Grove Museum of Natural History's website on the Museum to You page.